Part A. Analysis of a known mixture of group 1 cations, a positive control experiment. Prepare a mixture of group 1 cations by adding 15 drops each of 0.2 molar lead nitrate, 0.1 molar silver nitrate, and 0.1 molar mercury nitrate into a test tube. Then add 2 drops of 6 molar hydrochloric acid. Centrifuge the mixture from step 2 in order to completely separate the solid precipitate from the solution. Be sure you balance the centrifuge before you start. Add 1 more drop of hydrochloric acid to ensure that the precipitation is complete. If more precipitate is observed, then centrifuge the mixture again. Decant the supernatant solution into another small test tube and save the precipitate for later. The supernatant solution may be discarded into the lab waste container. Wash the precipitate with distilled water. Add 15 drops of distilled water then centrifuge. Prepare a hot water bath by adding tap water to a 250 ml beaker. Add water until the beaker is about 1/2 full. Once again, decant the supernatant solution into a small test tube. Save the precipitate and discard the supernatant solution to a waste beaker. Add 2 ml of deionized water to the test tube containing the clean precipitate and place it in the hot water bath. Periodically stir the mixture in the test tube. Remove the test tube from the hot water bath and immediately centrifuge the hot mixture. Carefully decant the hot supernatant solution into another test tube. save both the precipitate and supernatant solutions. In order to confirm the presence of lead 2+, add 1 drop of 6 molar acetic acid to the supernatant solution obtained. Next, add 3 to 4 drops of 1 molar potassium chromate. The formation of a bright yellow precipitate is confirmation of the presence of the lead 2+ ion. Add 10 to 12 drops of 6 molar ammonia to the precipitate from step 8. The appearance of a gray solid is confirmation of the presence of the mercury 2+ ion. Centrifuge the mixture and decant the supernatant solution into a small test tube. Save the supernatant solution now, since it contains silver. To the solution obtained in step 11, slowly add 6 molar nitric acid until the solution is acidic to litmus paper. The acidity can be tested by dipping a stir rod into the solution and then touching it with a drop of solution to a piece of blue litmus paper resting on a clean, dry watch glass. If the solution is acidic, it will turn blue litmus paper red, or it will have no effect on red litmus paper. Formation of a white precipitate of silver chloride in the acidic solution is confirmation of the presence of silver ion. Part B. Analysis and identification of group 1 cations in an unknown sample. Obtain a test tube which contains a mixture of group 1 cations. Record the ID code of the sample on your report form. And then add 2 drops of 6 molar hydrochloric acid. Continue following the same procedure used to analyze the known mixture of ions in Part A. Note that you may observe either a positive or negative result when you test for the presence of each ion, depending upon whether or not the ion is present in your unknown sample. On your datasheet, construct a flowchart similar to the one shown in the discussion section of this lab. Indicate on the flowchart whether or not the test for each ion is positive or negative. Then, in the space provided, indicate which ions are present in your unknown sample.